Hello everyone and welcome to Nanomedicine. I'm Professor Kimberly Kelly and I'll be your instructor for this class. So just to let you know, my office is in MR5, Medical Research 5, room 1214. Email to schedule an appointment or just drop by. If you're a biomedical engineering student or a nanomedicine student, you have card key access so you can get past the locked hallway. If not, then please again email me and I'll be more than happy to let you in. My email is kak3x@virginia.edu and I try to answer your emails as quickly as possible. Other ways to contact me are on Colab, so in the forum sites, and then also to give anonymous feedback. Please, with the anonymous feedback, please be respectful to each other and also to me. Everybody is working very hard, and so please, nothing nasty, just something very respectful, and I'll try to answer your questions or concerns. So the expectations in this class, on Colab, you will see the syllabus, and then also I have uploaded tips on how to succeed in this class. But expectations, the attendance is mandatory. Grading is not curved. If everybody in the class learns the material, then everybody gets an A. I'm happy to answer any question except what is on the test. For test review, come to class with questions. So when we have review sessions or review classes, I'm not going to reteach anything, and your TA won't reteach anything. This is all whatever questions you have, so please come with questions. In this class, you will use differential equations and other math. The math load is pretty light. It's not going to be like a computational class. But just to let you know, in years previous, there were people that were surprised that there was math in this type of class, but there is. So just to tell you that I do use the whole grade scale. Unfortunately, one student has failed the class, so but on the other hand, most students do really well in this class, especially if you participate, turn in all of your assignments, and actually come to class. So one thing I get on my evaluations frequently is that I'm unapproachable. I don't want that to be the case. I'm more than happy to take any of your questions or concerns or anything, so please don't be afraid to approach me. This is in general to everybody. We're all human. I was a kid once. At 2 o'clock in the morning before a half marathon, that's what I looked like, right in the middle. Nice face. And then I drink lots of Diet Coke and eat lots of peanut M&Ms, so my diet isn't usually the healthiest. But anyway, please don't be afraid to approach me. I'm extremely excited to talk to you. I'll talk to you about nanomedicine, the class, what your future plans are, what it's like to be a professor if you're interested in that. Anything that you're really interested in, I'm happy to discuss with you. So please stop by. I love talking to you. So let's get on to this part of the class. This is going to be a really short lecture. This is just an overview. So what is nano? Well, nano is the Greek word meaning dwarf. Nanotechnology is the creation and utilization of materials, devices, and systems that control matter on the nanometer length scale, i.e. at the level of atoms, molecules, and supramolecular structures. So it's very small. Nanotechnology, of course, is defined, and there's a National Nanotechnology Initiative, and it's the understanding and control of matter at dimensions of roughly 1 to 100 nanometers, where unique phenomena enable novel applications. So in nanomedicine, the high surface area to volume means that the particles have very interesting phenomena. A nanometer is roughly 4 times the diameter of an individual atom, and the bond between two atoms is 0.15 nanometers long. Proteins typically are 1 to 20 nanometers in size, so just to give you a rough comparison. Here's again a chart for natural things and man-made things about sizes. So ants are about 5 millimeters, atoms of silicon are 0.078 nanometers, ATP synthase from your biochemistry days is about 10 nanometers in diameter, and red blood cells, or cells in general, about 7 to 8 microns. So we're talking things that are pretty small on the DNA or the protein level. And then man-made things are nanotubes, electrodes, those are nano, self-assembled structures, X-ray lenses, pollen grains are very small. So these are some of the comparisons, just so you get an idea of how incredibly small we are talking about. These are little nanoparticles that are being phagocytosed by cells. So most cells anyway, can sample their environment by phagocytosis or pinocytosis. So they go out and they bring in, their membranes are not quiescent, they don't just stay homeostatic. Membranes are a very fluid thing. So cells will go out, they will encapsulate whatever's outside there and then bring them inside. And in this case, these are little nanoparticles. And what's interesting about them, if you see in B and C, this darker area from this electron micrograph is the membrane, the plasma membrane. And these aren't like little circles. These membranes actually function like shrink wrap. These phagocytosed particles are then brought inside the cell where they rest, probably perinuclear. So it's just an interesting thing. You don't even have to target these particles. They will be seen and be brought inside of the cell. Even things as large in G, for example, in micron sizes. So these are microparticles instead of nanoparticles. The cells are able to bring them in. You can see again these membranes coming around and reforming to bring those particles inside. So there's different characteristics and different flavors, if you will, of nanoparticles. In this class, you will be assigned to groups and you will be assigned to teach us about the different parts of the nanoparticles. Those templates are up on Colab, so if you want to check them out. But the different categories of nanoparticles are polymer-based, metals and metal oxides, biologics, liposomes or surfactants, and then also nanocages. So just a little bit about polymers. These are typically, again, polymeric structures, dendrimers, or PLGA, or other types of polymers that can chelate things inside and they form nanoparticles. Metals and metal oxides, so these are most famously represented by quantum dots. The interesting thing about quantum dots is that the fluorescence is determined by the size of the quantum dots. So these are metals and metal oxides because it's cadmium, selenium in the inside of the shell, zinc sulfide on the outside, and then a coating. And so they're really interesting materials. And then down here on the right is an iron oxide nanoparticle. So this is an MR image. Iron oxide will show up in MR imaging. And so this dark area is all the places where this iron oxide nanomaterial has accumulated in the aorta of this animal. So there's biologics. These are the viruses, or some people will call an antibody a nanoparticle. It is a nanoparticle, but it's the definitions. Biologics are interesting because they can be genetically coded. So this is synthetic biology. You can genetically engineer them, and they produce the exact same structures every time. Liposomes are lipid membranes. So these are lipolipid. These are lipids. They can form a single layer or a bilayer. This is more in keeping with what the plasma membrane looks like. And the interesting part about liposomes for drug delivery is that the inside of this, it's a hydrophilic side, so you can add DNA, RNA, or hydrophilic nanocrystals. In the middle is the hydrophobic region, so you can use lipid-soluble drugs or fluorophores. And then on the outside, you can put things like polyethylene glycol to protect them in the bloodstream. Or you can put targeting moieties like peptides or antibodies on them. So liposomes are really interesting structures. Here at UVA, I work on liposomes Rich Price from BME is working on liposomes and drug delivery, and also Sasha Klebanoff is an expert in liposome synthesis. And finally, nanocages. And so these are carbon nanocages similar to buckyballs. These are trimetaspheres, so these are, again, carbon nanocages that chelate or that have different metals or ions inside of them. So you can do imaging or you can do therapy with them. So what is nanomedicine? Nanomedicine is the application of nanotechnology to medicine and is based on three mutually overlapping and progressively more powerful molecular technologies. So nanostructured materials and devices, they are important for imaging, targeted drug delivery, smart drugs. Molecular medicine is genomics, proteomics, and artificially engineered microorganisms, again, synthetic biology. And molecular machines, for example, nanorobots for instant diagnostics, chromosome replacements, cell surgeries. Wearables have become really interesting right now. So the first application we'll talk about is drug delivery. In these nanomaterials, you can encapsulate them, these small molecule drugs inside of them, so you can get thousands to 100,000 more drug inside of these than having them in the small molecule. And this is especially important when the small molecule itself or the drug itself, when it's not encapsulated, is toxic. So if it was just injected or administered to the body, it would be toxic to the body. But this way, if you encapsulate it inside one of these nanoparticles, you can target it directly to whatever the disease is, and then it will accumulate there and limit the off-target toxicities to other organs. And so this way you can really change the profile of where it's being administered. We'll talk about these things in pharmacokinetics, and we'll learn throughout this course how to design the nanoparticles to get really good drug delivery. And also when you have to use this technique of encapsulation inside of a nanoparticle versus when a small molecule is perfectly fine. And then imaging is another application. So this is an example of a imaging agent, a nanoparticle imaging agent that is targeted to prostate cancer. So this is CT or computed tomography. And then this is a fluorescent imaging of imaging to prostate metastasis in this animal. So nanoparticles can be used for imaging applications. And then finally, something a little bit farther afield, but that is probably coming in the next decade, is nanorobotics. And so the ability for these nanorobotics to go into the bloodstream to find whatever is wrong and to fix it, either DNA in cells or other things, other applications. So nanorobotics. Something else that is coming up are wearables, and so that's a really hot topic right now. Google, I think, has been working on things, and they just released a contact lens that is able to detect, I think it's insulin changes or glucose monitoring in the eye. So that's all for this first lecture, and I will see you in class.